So let's look at the next hallmark of cancer, which is the evasion of growth suppressors. So what does evasion mean to us? Well, it means to escape. So to escape growth suppressors is equivalent to saying to allowing growth, right? When you evade something that inhibits something, then you allow the opposite process. So there are some key takeaways that we're going to go through, and those hopefully will give us a very good overall understanding of this topic. So the first one being the mechanisms of bypass. So there are many mechanisms of bypass, and bypass as in bypassing regulatory systems that are embedded within your cells. So this is our cell, right? We have our cytoplasm, and we have our nucleus, so which contains our genomic material, right, DNA. And there are signalings going on in the cytoplasm, and this can be again outside and inside out, and these are briefly mentioned in previous videos as well. So there are regulatory systems and signalings here that prevent excessive growth. So those are called regulatory systems that are embedded in the system of the cell, right? And then if we have the evolution of these bypass mechanisms, then there is evasion, right? Evasion of some of these regulatory systems. Now, to be more specific regarding this, we can look at some examples. So, first one being the loss of tumor suppressors or tumor suppressor genes, so TSGs. Now, TSGs, what they are, so in your DNA, so this is, for instance, your piece of your DNA, and you got a gene. So let's say this is gene, and this gene is a tumor suppressor gene. And actually, in your cells, you have 2 copies of every gene, so they're gonna lie parallel to each other. Now, if there is a mutation in both of these 2 copies of this tumor suppressor gene, then there is a allowing of growth. So then you can decipher what the function of these tumor suppressor genes are, as they are named, right? They suppress tumor. So they suppress, and then once both of these copies are mutated, then you have the allowing of growth. So there are many different different tumor suppressor genes in a given genome, as in the whole DNA of the cell that's located in the nucleus. So there are common examples, say P53, and there are many more, and we'll try not to get too detailed in there. But a very common one is this tumor suppressor gene here, P53, which is often mutated, right? Well, there are also sometimes tumor suppressor genes defined as having half to both of these copies being mutated, but there are instances where the mutation only on one gene copy is sufficient to give us a mutant phenotype, which is the allowing of the growth, probably in the context when there shouldn't be a growth. And that can kind of lead us into our second point here, is that evasion of growth suppressor involves inactivation of checkpoint controls. And what these checkpoint controls are, well, is a given cell, right? As we have most commonly drawn, they can divide. Each of these cells can then divide further. Now, this propensity to be able to divide really isn't available to all cells during the whole lifetime of a person. So a 60-year-old person, right, is not going to have all their cells dividing and dividing. This really occurs early on, but later on only specific tissues such as your skin, right, to replenish the dead skin will undergo this process, but not, or maybe some other areas such as your intestine as well. But so skin and intestine, some examples. But to undergo this, so let's consider just the first part is one cell dividing. There are many different checkpoints, so this is regarding this cell cycle. So there's the G1, there is the S, G2, and the M phase. And to go from each of these phase, from each phase to the next, which as a whole represent our cell cycle, well, there are checkpoints in each of these steps. Actually, there are two. There's one here, and there is one here, and there is one within the M phase. So these checkpoints, there can be more than one, and these regulate the cell cycle. And so if you have the inactivation of these checkpoint controls, such that you no longer, right, are controlling these processes, you undergo this cell cycle by a great amount. Of course, these are simplistic ways of putting all of this, that there can be interplay between various factors. Looking at the next note here, in the context evasion of growth suppressors, we have the dysregulation of apoptosis. So apoptosis, you may not have heard that apoptosis is equivalent to programmed cell death. Programmed as in it is not coincidental, it is intended. So intended cell death. And sometimes it is good for the cell. So if you have say the cell goes airy and it's really not that healthy for the cell itself to be able to undergo apoptosis, it's really good for the overall organism, because this is only one cell, and if it's abnormal, then it better, right, die than potentially contribute to something worse, such as cancer, by keep dividing and dividing. So apoptosis is programmed cell death, and there are many signalings within the cell that regulate, but more commonly is the mitochondria that has a role in releasing materials that such as cytochrome C, but that's a bit of a detail, that will allow the cell to undergo cell death. So denoted here. And the next note we're going to look at is the microenvironment adaptations that occur in context of tumor and in the context of evasion of growth suppressors. So again, if we have a cell, it is going to be in an environment, so in a context, right? So what is around the cell? The These are all factors that are going to influence how this cell and other cells are going to behave. So there's going to be other cells. They can be immune cells as well, and there can be proteins and molecules, right? So how those are regulated? Well, in the context of a tumor, it can really misregulate the surrounding environment and to better evade growth suppressors, such as a molecule. A molecule in the environment can be growth suppressor, but if the tumor cell is inhibiting it, then well, you have your evasion. So hopefully this gave you an overview. Of course, this is a very basic overview, and there is much more to this, but we'll stop it